What we're going to do today, and I'm going to refer to the words of George Bernard Shaw when he wrote somebody a letter saying, "I'm sorry I wrote you such a long letter. I didn't have time to write you a short one." And that is, we've asked our speakers to compress their talks into just a few slides, just to give us a refresher and to help us really see if there are themes that run through them. We hope that we have curated these talks well enough for there to be themes, and we do like, obviously, talks and presentations which are right out of left field from time to time. But we're looking forward to our speakers just to rerun for us what they've spoken about. Now, Alistair is our Director of Science. He's also visiting professor in quantum biology and bioenergetics at the Research and Centre for Optimal Health at the University of Westminster, where, of course, Jimmy is chair and Reese is coming in from Westminster. So I've asked Alistair to try and bring together some of the themes that that we hope have come out of the autumn series, and to then lead into the individual summaries and discussions from each of our speakers from this year. So, Alistair, can I hand over to you and keep it brief?
Thanks, Jeffrey. Yeah, thank you. I do apologise for this first slide. If your mind is going funny, it was not deliberate. Okay, next slide. That's just an overall overview. Really, what I just thought I'd try and do is just introduce a little bit of the background, how we got to where we are. As you probably know by now, we've always been interested in mitochondria. And this came about when Jeffrey, Jimmy and I started talking about this, because Jimmy and I were coming from lifestyle-induced disease and things like the metabolic syndrome, and why it is that a poor lifestyle seems to accelerate the aging process. And Jeffrey was obviously coming from the background of natural products. And we realized in about 2010 that many natural products, and this may surprise some of you, may not surprise some of you, that they fluoresce. Many polyphenols absorb in the UV range. And this was obviously quite interesting. And of course, then you start delving into the science behind this, and you realize, of course, many plant compounds are actually, or probably evolved, starting out as sunscreens to protect against UV. And in 2014, we started thinking about, well, how can we use this? And one of the questions we've always wanted to answer is that there's a lot of evidence to suggest that many natural products, and this includes some of the cannabinoids, seem to modulate mitochondrial function. But one of the questions is where are they in the cell? And so we got in contact with Stan, who's a whiz with lasers, and said, is it possible to track a fluorescent compound, like a plant compound, through a cell? Of course, obviously, we wanted to see it ending up in the mitochondria. Unfortunately, it didn't really work. What we did do was suddenly get a real insight and think, well, hang on, you know, what's going on with all the photons? Because what we did find, of course, is that shining lasers onto cells tends to kill them quite quickly if we have too much power. Anyway, we then started to try and image mitochondria using native fluorescence. And around the same time, we were also obviously getting into Nick's work. And in one of his books, he makes this lovely point about the potential across the mitochondria being the same as a bolt of lightning. Of course, this really got us thinking, linking the idea of photons and charge and everything. And so from then onwards, we started looking into quantum biology. And in 2017, we were lucky enough to attend a lecture by Mike on morphogenetic fields. And that really inspired us, because it was one of the most fascinating talks I've ever seen. And Jeffrey and I sat there going, wow, yeah, hang on, this is brilliant, because this is really it. And funny enough, a lot of people there didn't really get it, but we met up with Mike afterwards, and so we've been in contact ever since. And when we set up the foundation, of course, our aim was then, of course, we were very interested in photons and light. And so the first funded research was Westminster and Harwell, looking at photons and hopefully then electromagnetic fields. So this is kind of where we're coming from. It comes all the way back from fluorescence and medicine and what these plant compounds do in animals and humans, and why do they work at all. Next slide. Sorry, I'm pressing the wrong button. And of course, it's familiar to some of you. This is this concept put into practice, which of course, when we start looking at it, of course, it's all about, you know, where do electrons go? And you know, the idea being that, you know, can life be defined as an electron looking for a home? And when you look at electrons, of course, you start to think, well, they're very basic quantum particles or waves, which we like to look at it. And of course, the right amount of them is very good for capturing energy, but too many and you get a lot of damage, but just the right amount generates signals. So this suddenly begins to make the electron transport chain really critical in just about everything. And that little squiggle you can see there is a photograph I took many years ago of mitochondria. Yeah, thank you. With a dye. It's actually fascinating that when you start looking at it from that perspective. Anyway, next slide. So that led us on to our first autumn colloquium, where we invited a very eclectic mix of people. Philip Kieran coming from a completely different direction, Jimmy coming from the optimal health idea, Wayne, who was obviously into ATP synthase, and me a little bit looking at the quantum beat. But the main thing about this was we had all these very, very different people with different disciplines, but we all worked really well together. And Wayne in particular just kept saying to me, I've never thought of it from this direction before. And of course, it just showed the power of bringing these people together from very, very different backgrounds. Next slide, please. I won't go over this, sorry, this is just, I threw this up there. But this kind of is just trying to be a summary of, well, if we want to understand life and how it started, with the purpose of understanding what it's doing now, you have to look at all the various factors going right back from the beginning, and indeed back to the formation of the sun and the generation of compounds in interstellar space, back to the compounds on Earth, and how all of these things come together to generate what we call life. And one of the things, of course, we were very interested in, this comes back to some of Nick's ideas, is, of course, if thermal vents are very, very key, or were very key, or are very key in other planets in terms of generating life, clearly were quantum effects very important. We discussed this a bit before, but we also know that it seems that, as life is obviously obeying all the rules of quantum mechanics, then of course its next position is potentially the second law of thermodynamics. So we've got all the tools and everything we perhaps need to put together a bigger picture here. The next slide, please. And this led us on to the idea behind the theme for this series, which of course, based on the event idea, at what point do we start seeing quantum effects being really important in the origins of life, and obviously now, and what quantum signatures can we pick on this now? Okay, Nina, do we need to go through these next ones? Do you want to do them at the end? Because they talk about the outputs.
Great, Alistair.
I think great. We'll do those. If you do those at the end, Jeffrey, I think it'd be better to do those.
That's lovely, Alistair. That's sort of just a lead up to why we're all here now, and how we try to curate the spring and the autumn meeting. Thanks, Alistair.
And thank you, Al, for your beautiful summary, because I find myself well tuned with all this way you have been going through. In fact, On the Wave of Coherence was in my title, and especially something beyond the atomist point of view. Atomist point of view is very important. Is essential. I mean, you cannot do anything if you do not know what you are talking about. But then you have to collect all the pieces together. And this collection is made by coherence. Coherence is a dynamical law. Now, the starting point is indeed one of the points you were saying before, namely dissipation. Dissipation means that what we are talking about is embedded in an environment at any level. The single molecule together with the other molecules, a cell with the other ones, all together, and we are interacting this afternoon among us. So dissipation both ways. A trade of giving and taking. This is crucial and is beautiful if you can show that dissipation just use quantum field theory. I insist on this point, field theory, not just simple quantum mechanics, because in field theory, you have many quantum mechanics, if we could say in that way. You have many space of states, and they are different, physically different, technically unitarily non-equivalent. And when you go from one to the other one, that is a criticality. So the view we get is that a living system is a system which goes through continuous phase transitions through these states, these spaces of states. And this, there is a name for that. The name is spontaneous breakdown of symmetry. Here, the idea is that you start with something, an equation, a set of equations, which is your model, and there are symmetries attached to such equations. Now, when these symmetries are not the symmetry as dynamics realize them, then you say that symmetry is broken. It is idealistic, if you want the symmetry, which was manifestations came to us in some broken forms. And this happens because you can establish dynamically without, how to say, is not made by hand of somebody, but is intrinsic dynamics. You get long-range correlations among your quantum objects. Now, why quantum? Why not just classical? This is usual problem in biology. Why quantum? Well, if we have to deal with charge distributions, as we have to do at very low level, molecular level, then the only theory we have is a quantum theory. This is why. Then we have to get rid of quantum, trying to go to macroscopic level. And this is the main point of coherence. Since these long-range correlations establish coherent patterns, this coherence brings us to a collective mode. So the system appears as a macroscopic system. And this is a beautiful transition. This is not classical limit, sending H Planck constant to 0. No, it's something completely different. Just because H is not 0, you get macroscopic behavior. And this is ordered patterns, which are at the basis. So order is a lack of symmetry, comes because we are missing some symmetry. We can distinguish between things. And then, and then you have a construction of tissues, which is, I mean, a set of cells behaving collectively in with some functional activity. And then you have a lot of things. Next slides, please. Among those, you get fractals. Now, what we are talking about? We are talking about molecules, all of them made, characterized by some electrical dipole. So we have water, 90% of water. We cannot forget about water. And we have our big macromolecules or whatever you want. Now, how they communicate these things? Well, the picture we have in physics is that if you have two charges on the left side of these slides, you have two charges interacting. They see each other by exchanging a little ball, a photon. Similarly, in an environment of water, what you can exchange? Dipole waves. So this is the idea. If you can construct, as I was trying and I'm trying, and we have something, a formalism where you can describe interaction between macromolecules through polar waves, then you get a lot of things which I was telling you in my talk. Next slide, please. Now you get something beautiful, because a very general rule of quantum field theory is Anderson-Higgs-Kibble mechanism. I should say also Englert, Braut, and so on, because there were many of them. But most well known is Anderson in solid state physics, who discovered, actually, he was the one who discovered. Then we have Kibble, of course, and Higgs, of course. What do you get? You get the possibility to have self-control of the system. Because if you have an electrical excitation, which has to propagate through a coherent bunch of water molecules, then it propagates through filaments. It goes through little channels where you have E equal to 0, as you see on these slides, in this channel, And outside you have E non-zero because it's confined, is squeezed. The electric field, this is Higgs mechanism, by the way. The electric field is squeezed within this set of coherent objects. So you have a transversal gradient and you have a force. This force may attract or repel on the basis of a resonance force, whatever is around. In this way, you might test what you are doing, just let's say, by saying, let me see if I can compute, for example, how a microtubule is made and which one is the radius that I get. If you do that computation from such first principle, you get numbers like D1 equal to 146 angstrom for the internal radius computed in this way. And you compare with what you find on the biology books, which is 140 angstrom. So this is impressive. For a physicist, it's impressive. For a biologist, I don't know. But for a physicist, it's impressive because you didn't put anything inside except this dynamical mechanism and properties, of course, of water in this case, and of some number which are crucial number in all of it, namely Planck constant. And well, when the field propagates through channel, it gets massive. And the mass you get for such a field is the mass of the photon, which is electric field in this case, which is 13.60 electron volts, which is also this one is beautiful because it comes at ionization, hydrogen ionization energy. So this was the idea, the basic idea. I think there are no more slides. It's correct, yeah? Yeah, it's finished. So this is it. The idea is to have something to put together, some dynamical basic law. My proposal is that the dynamical basic law is one of coherence. Coherence comes before DNA. It comes before any proteic building of any large molecule. That is the origin of everything. Then you can work as usual you do.
Giuseppe, thank you very much indeed. That marvelous, and thank you for keeping to time. At that, can we go to Budapest, to Gabor, and receive your short presentation?
Hi everyone. It's very easy for me to continue after this talk because we also realized with Stuart Kaufman in 2010 that for quantum biology, coherence must be very basic, and we should understand what kind of molecules or other biological structures are responsible for conserving quantum coherence. So what is the best for a molecule to stay quantum coherent and enjoy perhaps the benefits of quantum mechanics, like solving problems much faster than in classical physics. And we also started from Anderson, who was mentioned in the last talk. Next slide, please. So in solid state physics, there is a description of conductors like metals. So in metals, you can imagine that electrons are just hopping on a lattice. And quantum mechanically, this is an easy situation. If you switch on the voltage between the two ends of a metal, it will just conduct, and the electron can go through it. If you add some impurities, then first nothing happens, the system remains a disordered metal, a disordered conductor, until it reaches a point where suddenly a phase transition occurs, and after this phase transition, so if there is too much disorder in the system, then quantum mechanical states become localized. And there is this underzone localization, the localization transition, and in the middle there are these critical states. And if you think about decoherence, if you add to the system some decoherence, so external noise, then what is going to happen? In the metal, you have extended states, and they are moving due to the external noise. And the external noise can destroy the quantum coherence very quickly because the system has long extended states which break very quickly if you add noise. What happens with the localized states? If you add noise, they are just tiny points and localized wave functions on tiny points. They just move with the noise and nothing happens. They stay coherent, but the system is not very interesting because biological systems have some functions, mostly transporting electrons from A to B in some biochemical process. So neither the metal nor the localized limit is helpful for biological function. And it turns out that exactly or very near to the critical point, so if you have just the right amount of disorder in a system, then you have fractal wave functions in the system. I can go back to Giuseppe's talk, so fractals seem to be very important. And these states share the resistance to external noise with the localized systems, but they share the transport properties with the metallic system. So they are at a pot where you still have transport, you still can have very good transport, but your system is capable of staying quantum for a long time. And in solid state physics, there is a prediction. I mean, it's easy to look at a system and foretell whether the system is quantum critical or extended or localized. You just have to look at the quantum mechanical energy levels and you have to make a statistics out of it. And so out of blue in 2010, we had this idea that drug molecules should have critical states, and we also patented this very nice idea. And it took another 2 years when we met in Canada Denis Salahab, who is a very well-respected quantum chemist, and we asked him to calculate for us the spectra of some biomolecules, first small ones. And next slide, please. What came as a surprise that without any tuning, without any other thing to do, we just got small molecules to even large proteins, the energy spectra, and without any tuning, they were really sitting on the critical line. It's very difficult in solid state physics to artificially produce such quantum states. Solid state physicists like these states because they support, for example, superconductivity, they support electron pairing. There are lots of interesting things which they cannot measure because they don't have such special materials. And here it turned out that in proteins, And in biomolecules, this is really just there without any tuning. And then when we published this paper on the level spacing of proteins and smaller molecules, some experimental groups reached us first from Arizona State University, where Stuart Lindsay measures long proteins, and he finds that the proteins have almost zero resistivity. So the protein itself is really close to an ideal conductor, and all the resistivity comes just from the metals his apparatus is connected to the electrodes. And right now we are working with another group at the Weizmann Institute of Science with David Cahen, who measures in another setup between two electrodes the conductivity of proteins, and we dug out 10 years of data from Weizmann Institute, and we now can show that all the data they collected and published, and they had different ideas what's going on, boils down to a single idea that all the resistivity is on the metallic contacts they use, and the proteins inside conduct almost perfectly, like quantum mechanically. Next slide, please. And why is this important? Because all the receptors in biological systems are made of these proteins and the packets in these proteins. And so far, drug discovery worked, by just checking whether a molecule a ligand can fit into the receptor. And after that, they sort of, if the molecule was fitting and filling the hole, that was the basis of the drug discovery. But now we start understanding functionally what happens if we put a small molecule, because on the protein there is charge separation, and these small molecules, like the key in a hole, fits in and connects like these magnetic keys, the right points in the protein, so that the charges can flow and the protein can change its shape and it can carry out its action. So there seems to be a relevance to this, and we also think, next slide please, that actually this has a very strong evolutionary basis, that because not all molecules have these properties, only molecules which take part in electron or charge processes in biological systems. So we have a strong feeling that already at the beginning these molecules have been selected for this task by evolution or whatever, to carry out these functionalities. Thank you.
Gabor, thank you very much indeed. That's excellent. Thank you. Can I now ask Greg to present his slides?
Great, thanks Nina, Jeffrey. Yeah, so I'll just remind you quickly of my talk, and I'm going to focus on the part of it that seemed to get the most discussion. So Nina, could you please change the slide? Yeah, this is just a reminder that I spent a lot of the talk talking about experiments on photosynthetic proteins with short laser pulses. And the bottom line of all of this was that we could use the short laser pulses to prepare superpositions, coherences of different kinds in these systems and watch what happens to them. And the message was we can prepare these coherences and they're passive in the sense that really they just get beaten up on by the environment and disappear. And so they last for maybe picoseconds at most. So that led me to wonder, you know, if we're going to have functional coherences in such complex systems as biology, they need to not get beaten up on by the environment. So how could that happen? And the next slide, please, Nina. So the next slide is inspiration from hundreds of years ago on classical systems, nonlinear oscillators that can do the opposite. So we can start them out of phase, the pendulum on the two clocks here, and through very small interactions, basically feedback, the same as an engineer might use, they can come into phase. And so this is the exact opposite of what we saw in those other coherences. So they're engineered to be robust. And this is not violating the second law or anything like this. In fact, entropy does increase. Oh, just a sec, hang on, just a minute. We could go to the next slide, please, Nina. So that led me to wonder more about synchronization. Now, it can be really powerful, and the key is it's robust. So there's many phenomena. This is, of course, abstract statement, but there's many examples where very weak interactions can lie at the heart of collective phenomena. And at some critical point, huge number of these independent systems essentially condense to produce new function from synchronization. So in the case of these coupled oscillators, you can have thousands coupled in theoretical models, and there's a phase diagram for them where they can be completely disordered, unsynchronized, and then with certain threshold, they'll synchronize. And you know, this is not necessarily quantum, of course. These models I'm discussing are classical, but they seem to me very practical and something that almost certainly has to be part of the puzzle here of where coherence would come into complex biological systems functionally and robustly. And I think there's one more slide, Nina. Oh no, maybe there's two. So the question is, instead of looking for pairwise interactions that are large, that sustain the coherence, you know, and not let the environment, as I said earlier, beat up on it, maybe what we need to look instead is for networks or clusters, systems that collectively produce independently these robust states. So it's the opposite point of view in many ways. And then there is the last slide, which is just discussion topics. You know, I think there seems to me to be a lot of scope for understanding more about these collective phenomena, what makes them powerful fundamentally. And there's some interesting physics and mathematics there, and of course they have been studied extensively in these nonlinear systems for many, many years. But I think there's a lot of scope here. There's evidence that these kinds of collective effects are seen in brain function in experiments I'm talking about here, not simulations, where there seems to be periods of disordered brain signals that somehow all of a sudden coalesce into a conscious thought. There's some papers on that conjecture this, but based on experiments. So this could be more widespread, of course, in biological systems. You know, where does apparent engineered like function come from in cells. It's feedback control loops at the core of it, but it's much more interesting than that, I think. The third point is, are they quantum analogs, which they can be. These systems, are they relevant to biology or not? It's an open question. Okay, and finally, of course, well, given these conjectures, what next? What should be focused on? So, thank you.
Greg, thank you very much indeed. Ably assisted, I see there, by your junior research assistant.
That's right.
Lovely. Thank you very much, beginning to draw the link between the earlier two discussions and onwards into biology. Can we now ask Marco if, Marco, you're going to share your slides, I think.
Yes, sure. Okay. Can you see my screen?
Yeah, we've got them. Perfect, thanks.
Okay. So quickly, what I've been talking last time is Freilich theory. In other words, when you have a macromolecule at thermal equilibrium, all its atoms or subcomponents make incoherent vibrations caused by the heating of, say, water molecules. But if you, so at thermal equilibrium, but if you inject energy, and of course, you need some receptor and some source of energy. This could be ATP hydrolysis, photons, biophotons coming from redox reactions, whatever. If you inject energy, there is a phenomenon, a collective phenomenon, which can be switched on if the injection rate exceeds some threshold value. And all the parts of a macromolecule can start moving coherently and making a giant oscillation. And this could bring about a giant dipole vibration. And as Giuseppe was mentioning, the role of water encircling these molecules is very important in enhancing the value of the dipole moment. What Freilich suggested was that if you have giant dipoles oscillating at resonance, you can establish long-range interactions of electrodynamic origin, of course. If they are not at resonance, you have a potential, an interaction potential which falls off definitely quicker. And what I told you is that by attaching chromophores to a protein and shining a laser light on this protein with these chromophores, we have been able to find actually this condensation phenomenon of the vibrational modes. And it was also done with red phycoerythrin, a protein which is naturally expressed with many chromophores. Nature selected it to collect light. And okay, by means of experiments, we found in agreement with the theory, with the back on the envelope computations in much more refined models, we had the confirmation that actually we have been able to excite these collective modes of a protein. And this is at the origin of the possibility of getting long-range electrodynamic interactions. Long-range means that we have found that identical proteins, so vibrating at resonance, because the resonance is built in, they can interact, they can see each other at distances larger than 1,000 angstrom. So, which is rather long with respect to all the other dispersive forces acting around 4 or 5 angstroms. And I told you that, okay, there is an interest of this phenomenon in the study of the relationship between brain and mind, essentially by referring to what has been proposed by Roger Penrose, who reflection on the roles played by microtubules inside neurons, and all these microtubules inside neurons form a so to speak, hyper network, and Roger Penrose and Stuart Hameroff suggested that this kind of coherence phenomena, like the Fröhlich condensation, could be at the origin of elementary phenomena that are interpreted as a moment of consciousness. I don't want to go through everything, just a quick recall. If the tubulins forming microtubules have two states, and if you consider the possibility of having these molecules in superposition of the two conformations, thanks to a mechanism like the Fröhlich condensation, we can have a larger and larger number of molecules entering in resonance and in a coherent state, and all of the sudden, when the mass of a sufficient large number of molecules enter in this coherent state, then there is a sudden wave function collapse, and that is identified as a moment of consciousness, an atom of consciousness. Okay, I've shown this. And what I didn't tell I told you last time that, of course, tubulins are heavy enough, and so this proposal have been questioned because people say that you cannot have superposition of states of such a heavy molecule, but the water molecules inside microtubules can support the coherent phenomena, which in quantum optics are called pi pulses, and the aromatic rings that are inside these proteins can form regular lattices, which are called pi stacks, and these can actually work quantum mechanically. So we have three levels in interaction: a classical one, so to speak, and inside microtubules and at the interior of microtubules, the coherent states that can be induced in water molecules, and in pi stacks are quantum phenomena which are under investigation. What I didn't tell you last time is that there is a lot of experimental evidence and even hospital trials concerning the effects of electromagnetic fields on cancer cells. In other words, the action of external fields, magnetic or electric fields, very weak with respect to what one would expect from a classical point of view, are capable of stopping the mitotic spindle and are capable of stopping the growth of cancer cells. And there are clinical trials and experimental evidence on this fact. And of course, if we better understand how macromolecules like proteins interact through electrodynamic, long-range electrodynamic interactions, if external fields are capable of putting them off resonance, for example, or putting them in resonance if they were out of resonance, perhaps we can better understand why electromagnetic field can have this healing effect on cancer cells. Okay, this is more or less.
Marco, thank you very much indeed. Okay, Nick, over to you.
Thank you. So I suppose I'm going to give a very brief overview of what I said, but more I'd like to try and put it into the context of what other people have been talking about. My talk was about the origin, really, of membrane bioenergetics, where it all came from in the first place at the origin of life. The striking thing is that all cells rely on membrane bioenergetics, and this is the aerobic system in bacteria. You won't be able to see my cursor, I don't suppose, but the main point there is it's a very complex system involving multi-subunit proteins. I mean, the one at the left there, complex 1, has about 45 subunits in our own cells, in our own mitochondria. There's two potential quantum processes going on, as I see it. I'm not a quantum biologist at all, but one of them is linked to quantum tunneling, which is what I was talking about in my talk. Electrons hopping from redox center to redox center. It's been under selection for distance all the way down that respiratory chain. You're probably going through about 25 separate redox centers from one end of the chain to the other, something of that order, and they're all under selection for distance. The other aspect that came up in discussion, and that Alistair in particular raised, is interactions with the conjugated electron systems. Now, quinones have them, flavins have them, cytochromes have them, and so on. So they all interact with light. And so I have the feeling that Alistair, at least, and perhaps a few people, would like to see the potential for quantum interactions in generating proton gradients at the origin of life. Now, I've seen it the other way around, I have to say, because I cannot understand how such a complex multi-subunit interacting system, this is the old chicken and egg question, comes about in the first place. So there's...
Nick, you've hit your space bar, I think.
Sorry, could I hope you didn't miss much of that. Could I have the next slide, please? Okay, so this was what I presented as about the simplest system. There are geological environments where there are natural geologically sustained proton gradients across barriers. And so you don't necessarily need to generate your own proton gradient to power work in cells. Now, this is a methanogen on the left-hand side here. The protein in the membrane, the cylinder in the membrane there that's marked ECH, is the energy converting hydrogenase. That's a proton motive protein, membrane protein, which is to say it's drawing on the flow of protons down a concentration gradient and an ion gradient, an electrical gradient, to reduce ferredoxin, which is an iron sulfur protein. The ECH itself is an iron sulfur protein, so we're dealing with iron sulfur clusters, protons and electrons, so we're into quantum territory immediately in thinking about these things. There's kind of a switch. If you look at the right-hand side there, I'm showing the kind of very schematic structure of the energy converting hydrogenase. The problem here is that under the conditions in which cells operate, the reduction potential of hydrogen at around about 10 pascals pressure is about minus 300 millivolts. And it's reducing ferredoxin, which is in bacteria predominantly in the reduced form, so it's about 99% reduced. The reduction potential there is about minus 500 millivolts. So we're a 200 millivolt shortfall, and yet that goes ahead, no problem. It goes ahead because of the proton motive force. And I showed possible mechanisms, which I imagine rather than know. There's no experimental demonstrations of this to my knowledge, but the reduction potential of all of them depends on pH, just by the standard Nernst equation. Two of those iron sulfur clusters in there are pH dependent. So this is basically a redox switch driven by proton gradients across membranes that can allow ultimately CO2 fixation. Now, to me, that's the single most important thing that you need to do at the origin of life. You need to go from a purely inorganic world with gases like hydrogen and CO2 to organic molecules that will self-organize. For example, you will get bilayer membranes from fatty acids, and you can make fatty acids from hydrogen and CO2, and they will form protocells and so on. So the most important driver that you need is growth. And from that point of view, heredity itself is just a form of growth. Copying DNA means you have 2 DNAs where you used to have 1. So heredity is just a form of growth. Growth has to come first. Growth is powered by ion gradients across membranes, and those ion gradients necessarily come with fields, as we've all been saying. Now, sorry, could I have the next slide, please? So this is the kind of hydrothermal environment that I was talking about. In the middle there, that's Lost City hydrothermal vent. You can see next to it, there's basically a kind of a labyrinth of interconnected pores. The hydrothermal fluids themselves are strongly alkaline and rich in hydrogen gas. The ocean fluids on the early Earth in the Hadean would have been mildly acidic, perhaps pH 5 or 6, we don't know for sure. They also would have percolated through this labyrinth of pores. Now, on the left-hand side here, you can see 2 effectively testable hypotheses for what might be going on. So A, at the top there, we have hydrogen at pH 11. That makes it more reducing in an alkaline environment. A semiconducting iron nickel sulfide barrier. So the electrons come from hydrogen, reduce the barrier, cross the barrier, and then reduce CO2 to formaldehyde or organics generally. That's really supposed to be organics generally at pH 6 on the ocean side. If you follow that through to its logical end point, and I'm not suggesting that this necessarily happens, there's all kinds of things you could imagine, but the B then is effectively, if you've got organic synthesis happening there and nothing goes away and it self-organizes as you would expect it to, then you would end up with bilayer protocells on the acidic side of the channel. Now, an alternative way of seeing this in C is that it's not the electrons that are crossing the barrier, it's actually protons that are crossing the barrier. And so we then have a very steep proton gradient at the alkaline side, which is capable of reducing hydrogen. Again, the hydrogen is strongly reducing in the alkaline conditions. The CO2 is still in acidic conditions, but right at the very edge. And I'm showing roughly at the right-hand side there what I have in mind. And then the logical endpoint of that is protocells in the alkaline channels with a flux of protons coming in, which is topologically equivalent to what we have in modern cells. That's my currently preferred option. I showed some data suggesting that you do have very steep proton gradients. Sorry, could I have the next slide, please? But we also did some simple modeling, and I didn't show this slide before. This was never intended to be shown to anybody. This was just really tracking our own ion movements in the model, and I mentioned it in the discussion and wanted to show it to you. So the model I did show you, it's in the middle there, we were just tracking the movement of protons from an acid channel at the top to an alkaline channel at the bottom, going through a leaky protocell with proteins in the membrane, so we could control the permeability of the membrane, we could control the speed of proton flux through the proteins and so on. We used pretty standard classical equations. None of us are physicists. And what I'm showing at the left and the right-hand side is the free energy and the membrane potential, the delta psi. We have the ocean interface and we also have the vent interface there. What is interesting to me is that we do get a delta psi, even though there is even at time 0. And the reason for that is in the Goldman-Hodgkin-Katz flux equation, it depends on permeability to these ions as well as the concentration. So we can set the concentrations and we can balance the concentrations and balance the charges, but the permeabilities are different. And so we end up with a non-zero delta psi at the beginning. That's probably an artifact of being too simplistic in the modeling. If we introduce more ions, that would disappear. As protons come in, we get a positive delta psi inside because they move in faster than any compensating ions. And that's exactly the opposite way around to modern cells. So really what I wanted to show you at the right-hand side here, if you look at pH, so about halfway down, no, sorry, I don't want you to look at pH, I want you to look at delta psi 0, that's the second one down, in the presence of 5% pump. So you can see that delta psi is becoming more positive in the absence of a pump in the left-hand panels, but as soon as you have a pump, it's becoming more negative inside. So there's a flip as soon as you add a pump. And what that means from the point of view of these, either way, we have fields. As soon as you have any kind of protocell, we have fields operating across the membranes as a result of continuous flux of protons coming in. As soon as you introduce pumps of any sort at a later stage, you flip that field. I have no idea if that's significant, if it's meaningful in this context or not. But as soon as you have a pump, then you're generating a field which is acting on the cell as a whole. And to my mind, that is the origin of electrical fields in biology. We're talking way before DNA. We're really talking at the earliest levels of organization in generating these fields and holding a unit of the cell as a unit, as an individual, let's say. Can I have the final slide, please? Okay, so this is a bacterium being attacked by viruses. There's a couple of things I just would like to, points I'd like to make about bacteria. The mitochondria that we've talked about a lot, of course, derive from bacteria in the first place. Their membrane potential is the equivalent to the plasma membrane potential in bacteria. And if a bacteria gets attacked by viruses, for example, the best thing that it can do, and there's some very beautiful work from population genetics showing this, the best thing that it can do is kill itself before the virus has a chance to make any copies of itself. Because if it can make copies of itself, then it will infect the neighboring bacteria. And so to prevent its kin cells from being infected by the virus, and there has to be very little genetically in common, basically any other cell will do, it will share some genes, it needs to kill itself immediately. The way it does that is by plugging in a pore into the membrane and collapsing the membrane potential. In other words, this life or death for a bacterium is about membrane potential, and it's collapsing that membrane potential. And of course, I think Alistair mentioned, what about the battle with viruses as well? Viruses will try to prevent them from collapsing their membrane potential. And so there is a battle going on over life and death in bacteria between viruses and bacteria about the control of the fields. Now, the other thing, which I actually only just recently came across, but talking about morphometric fields, bacterial cell division, the bacterium needs to find its middle. And then they form a kind of constricting circle around the middle, which divides the cell in two. Now, most of those proteins, their position and the way they find their position, there's quite a large literature on oscillating proteins that go from one end of the cell to the other, but they get lost if there's no membrane potential. So if you depolarize that membrane, then those proteins disappear and end up dissipating throughout the cell, and the bacterium is no longer able to divide or have any idea where it is. So this is not only morphometric fields in planaria and so on, morphometric fields in bacteria as well. I think most people, when they're thinking about the proton motive force and membrane bioenergetics, are thinking about ATP synthesis. I suspect that the reality is we're talking about fields, and that the field, there's two reasons that I've just given you there. One of them is it integrates the bacterium as a unit, and the other one is it tells you where things are inside it and where they should move to power these processes. So this is something which arose right at the origin of life. What this flipping of charges might do as soon as you start pumping, I'm not quite sure. I'm going to stop there because I think it's better to discuss it rather than keep talking. But thank you.